Hello, everyone, and welcome. My name is Jaymar. I'm a senior marketing associate here at Nanion Technologies, and I'm happy to be your host for today's webinar. Supporting your drug discovery research with the SURFE2R platform. And for that, we have a special guest coming from Sygnature Discovery, Dr. Adriana Bizier. Joining me as the moderator of today's webinar is my colleague, Rocco Serlotti. He is an application scientist here at Nanion Technologies, and he is part of the SURFE2R team. He will start with a brief introduction, and later he will be moderating the Q&A session. So it's my pleasure then to leave you with my colleague, Rocco.
Hi, everyone. So as Jaymar told you, I will be the moderator for later on for the question and answer. So please write the question in the Q&A box. So now I will just share my screen. I will not keep you too much. I just want to talk briefly about the technique itself. So solid supported membrane-based electrophysiology, as the acronym says. We can start by saying that our sample, so we collect our sample by breaking our cell or native tissue. Then from the fragments that we obtain, we can either apply sucrose gradient centrifugation and collect the separate membranes depending on their density, or you can also purify your protein and then reconstitute into proteoliposomes. Anyway, the good things about the sample is that it can be stored at -80 for whatever time you need, and then it can be used on the platform for the measurement. So those are the two platforms that we have here at Nanion. This one is the low throughput one, so only one sensor per machine, and then we have the high throughput one, 96 sensors, so 96 well played, so 96 recording at a time. Okay, so how do we start? We start with our gold surface sensor here, and we functionalize it with an artificial bilayer that is composed by a thiol and then a lipid layer on top. So on this sensor, we let our sample absorb, so again, membrane vesicles, organelles, or proteoliposomes with the protein of interest, and then we put everything inside our machine, so it's connected to an amplifier, and we can have an electrical readout. When we are in this, when the sensor is in the machine, then we can perfuse our solution containing the substrate. We start the measurement at 0 voltage, and then thanks to the protein transport activity, we will have an accumulation of charges inside the membrane. The whole system acts as a capacitor, so we will read a capacitive current, which means we will start with a membrane voltage of 0 here, then we will have a capacitive current. At the top here, we will have, at the top here, we have that the capacitor is fully charged, and basically the current now starts to drop until the membrane voltage reaches maximum and the current is again 0. This is just a brief lookout on how a normal workflow on the instrument look like. So we have a continuous solution perfusion here, 3 solutions to be precise. So a non-activating solution that does not contain the substrate of interest. This will get rid of the mechanical artifact that comes into play when the fluidic is set into motion. We get our baseline for the measurement. Then we have the perfusion of our activating solution. This time this will contain the substrate of interest, and we hopefully have our electrical readout of the transport that is happening. And in the end, we go back again to the non-activating solution, so the transport will work in reverse, and basically we set the system to the initial conditions. So now it's my pleasure to present you Adriana Bizier, She did her PhD in biochemistry and microbiology at the University of Strathclyde in Glasgow. And her expertise is on protein purification, protein malfunction in diseases, and of course, electrophysiology. She currently works as a senior scientist at Sygnature Discovery in Glasgow, and she is specialized in assay development on the SURFE2R platforms to help accelerate compound screening campaigns. So I would say that the ground is yours, Adriana. Please feel free to go.
Thank you, Rocco. Let me share my screen. Hello, everyone. Thank you for tuning in today. As Rocco mentioned, my name is Adriana and I work at Sygnature Discovery. And today I'm going to talk to you about supporting drug discovery with SURFE2R platform here at Sygnature Discovery. But first of all, allow me to introduce our company just a little bit. We are a contracted research organization with over 900 scientists spanning 4 sites in Canada and 4 sites in the UK. And our services, the services we provide with our clients with begin with target identification to validation, all the way through to candidate selection. Our drug discovery expertise, both integrated and standalone services, span across multiple disciplines. Those include protein expression and structure biology, in vivo pharmacology, DMPK and formulation, as well as chemistry, and spans across different therapeutic areas, such as oncology, neuroscience, immunology, metabolism, and infectious diseases. But today I would like to turn your focus to our side here in Glasgow in the UK, cellular pharmacology, formerly known as SB Drug Discovery, where we're specializing in supporting early stage drug discoveries from target-focused cell line generations and assay development all the way through to high-throughput screening and lead optimization studies. Our 20 years worth of experience translates to over 500 different assays for different targets, utilizing wide range of platforms such as fluorescent-based flipper assays as well as electrophysiology assays. However, recently our attention was turned to SLC transporters, where we looked into developing assays to support drug discovery campaigns with our ligand binding assays, fluorescence-based screening assays, radio-labeled uptake assays, and finally, state-of-the-art SSM electrophysiology, which will be the main focus of today's talk. But before I start talking about SSME alone, I would like to introduce you to solute carriers, or SLCs for short, transporters. It's a family composing over 300 membrane-bound proteins that facilitate the translocation of a wide array of substrates across biological membranes. Those transporters play a crucial role in physiological processes, such as cellular uptake of nutrients or absorptions of drugs. Human genetic studies have provided insights into the roles of SLCs in both rare and common diseases, suggesting a wealth of new therapeutic opportunities in this area. Indeed, several classes of marketed drugs target well-known SLCs. However, lack of appropriate tools for direct, label-free and high-throughput screening assays are still a bottleneck for drug discovery campaigns and development of new drug targets targeting those transporters. This is where solute-supported membrane electrophysiology comes into play. It offers a reliable method for the measurement of ion transporters, overcoming said obstacles. So I'm not going to spend too much time going over details on how the technology works, because Rocco did a great job explaining it, and he's obviously an expert in this. But very briefly, solute-supported membrane electrophysiology is supported by the SURFE2R platform developed by Nanayan, and is ideal for studying electrogenic transporters. It measures net transport of charged ions. The advantage of SURFE2R technology, as Rocco mentioned, is that no continuous culture of cells is required. We simply take purified membranes, and this is just a simplified schematic what it looks like, subject our cells, release them and subject them to a series of centrifugation steps to then extract plasma membrane fraction to look into the proteins that are specifically expressed there, or organellar membrane fraction for your lysosomes, mitochondria, and so on. As Rocco mentioned, a good thing about this is that the sample can be stored indefinitely in the -80, and you can grab it at any point and do measurements without any significant loss in signal itself. That means it provides a high stability, and multiple repeat experiments can be performed on one batch of purified membranes. Before I go and dive deep into the case studies that I prepared for you today, I would like to highlight to you how important the sample quality is for the success of the SURFE2R assay. And hopefully I'm going to convince you by showing you a couple of examples of what it might be and entail. So first of all, we looked into purification methods and compared them on the SURFE2R platform. And here I'm showing you an exemplary figure showing EAT2 L-glutamate translocation, where we compared high-speed centrifugation method versus density gradient centrifugation method that was shown on the diagram on the previous slide. You can see that the density gradient improved the current signal by almost 5-fold in comparison to just high-speed method centrifugation. We also checked different expression methods. We tested membranes from cells stably expressing the target, in this case, again, EAT2, versus membranes from cells transiently expressing the target. And for that, we chose HEC293T expressing EAT2 cells and COS1 cells, again, expressing EAT2 cells. As you can see on the average, I should also note that all these samples were standardized before were applied onto the surface of the sensor. So the protein content was constant across all of the sensors, regardless of which sample was tested. So on average, membranes coming from the stably expressing EAT2 yielded a higher average response. However, you can also see that the transiently, the membranes from the cells transiently expressing the target are also giving a very decent signal. At the end of the day, it's all matters is that we have a good assay window between the untransfected. In this case, I'm showing you HEC293A parental cell line. And as you can see, all of them are giving a pretty decent signal. The only difference being that with stably expressing cells, we have somewhat a better idea on the expression levels, and they are more consistent across the batches. So with that in mind, now I'm going to go into our first study, case study, which is EAT3. And it's a sodium-dependent excitatory amino acid transporter belonging to SLC1A family. It is responsible for extracellular glutamate removal in central nervous system. Its impaired function in the body has been proposed to be involved in diseases including Alzheimer's, Parkinson's, and epilepsy, obviously making it quite an interesting target to look at pharmacologically speaking. And here at Sygnature Discovery, we have both platforms, SURFE2R 1 and SURFE2R 96, that you would have seen on Rocco's presentation. And how we normally begin with assay development, we usually start on SURFE2R 1, simply because it is more flexible. You can stop it at any point and reset your conditions and start all over again. And it's not consuming nowhere near as much sample as SURFE2R 96. So the first question that we usually ask when we begin with the assay is there an assay window? So for that, we tested multiple sensors with EAT3 on them and exposed it to L-glutamate, in this case 1 millimolar, and repeated the same conditions on the untransfected HEC parental cell line. You can see the window is pretty good. With that in mind, we ask ourselves, is this the response dose dependent? For that, we repeatedly expose sensors to different concentrations of L-glutamate and plot the results, average results, using PRISM, where you can see that the EC50 value was about 140 micromolar. So yes, it is indeed dose dependent. Then for further validation of the assay, we look for any available tool compounds commercially available. In this case, it was WEI213613, which is a known inhibitor of EAT3. And again, we exposed EAT3 translocating L-glutamate to increased concentrations of whey compound. And as you can see at the highest concentration, which is 50 micromolar here, the current is entirely abolished. And the IC50 yielded was about 9 micromolar, which fits quite nicely with what's been reported in the literature. With this in mind, we can now scale everything up to 96-well format for more data. As it usually happens, there's always some things you have to improve when transferring from one platform to another. And first thing being is establishing proper membrane dilution. I should have mentioned that when you purify your sample, it's pretty concentrated in not only your target, but also other proteins. So what you want to do is avoid overloading the sensor with too much of the protein or having not enough of it. And for that, we always establish a proper dilution. That also avoids burning through your stacks of sample quicker than necessary. After that, we always look at the assay conditions, if there are any improvements that need to be considered. And this is a graph showing initial conditions that we used for EAT3 on SURFE2R platform. As you can see that in the excitatory period or your own signal where your substrate will be applied, the baseline, which is just a buffer, non-activating buffer alone, here in black is shifting quite a bit to a negative value. So after a series of adjusting the pipetting speeds as well as the height of the tip before the liquid is dispensed onto the sensor, we get our final conditions where, again, in black, you see the baseline where it is quite steadily and consistently at 0, sitting nicely at 0, and where your excitatory period is, or your own signal, is also sitting quite nicely. And we're quite happy with those conditions. Then we look into assay and sample stability. For that, we kind of try to mimic what we would normally do, kind of layout of the deck, mimicking compound addition. So here I'm just showing you steady L-glutamate concentration across all the liquid periods. And we stack traces from the 4th liquid period and 7th liquid period, because that's where our compound would be. And as you can see, there's no much difference between the two. The shape of the peak as well as the height remain the same, meaning that switching between different reservoirs doesn't affect the conditions and is pretty steady, as well as the sample being stable over repetitive exposure to fluidics exchange. To also even further mimic what would happen with a compound addition, we introduce incubation period, as some of the compound might require an additional extra time to evoke any effect on your target. And as you can see, that there's absolutely no difference. It's a nice steady line indicating that those conditions are satisfactory. With that in mind, we then were able to generate a lot of data very quickly. And as per usual, we look into reproducibility. And this is a graph showing the dose response of L-glutamate in EAT3 over 3 separate days. And the EC50 was consistently sitting around 130 micromolar L-glutamate. We then went on to validating our assay in 96-well format using inhibitor. In this case, I'm showing you here DLTBOA. And allow me to spend a little bit more time on the graph here, as this would be the normal way we show the our results and how we analyze them. So we usually start our workflow with the baseline. This is just addition of your non-activated buffer, so there should not be any activation present. And this is just to exclude any instabilities or mechanical issues. Then we move on to our control current. Usually for that, in our inhibitor studies, we choose EC80 of the substrate, in this case, L-glutamate. Then we move on to the control current, so exact same buffer, but this time in presence of the tested compound. And as you can see, I have a red box around the highest concentration of the DL-TBOA. It doesn't really look like it's going to full inhibition, but when you look at the raw traces and data, you can see that the control current, so from your period where compound is absent in black here, when overlaid with the full antagonist concentration, so DL-TBOA, it produces nearly flat line, meaning that it goes into full inhibition indeed. And IC50 that we got from this study was about 2.6 micromolar, which corresponds quite nicely with what's been reported in the literature for this compound in EA3. Having EA3 assays successfully established on SURFE2R platform, we managed to translate those and apply those conditions to other membranes of the SLC 1A family, including EA1 and EA2. We validated these assays with DL-TBOA as they all being inhibited by this compound, and the results were pretty consistent with literature. Another thing I would like to talk to you about today with regards to SURFE2R is how flexible the assay is. SURFE2R platform allows for a very swift change between tested buffers. So you can test multiple substrates, endogenous substrates, on the same sensor or well, depending which platform you're using. And you can also swiftly change between tested pHs. And you can go to quite low pHs without affecting the sample because it is a membrane, as opposed to a live cell, which would normally not like being exposed to a very acidic pH or a very alkaline pH. And the latter is quite important for some of the targets which heavily rely on the pH for their optimal function. And examples are SLC36As and SLC15As. So the first example from SLC36A is PAT1, belongs to a family of proton-coupled amino acid transporters. It is being identified as a drug carrier, mediating absorption across of intestinal luminal membranes, and it's involved in membrane transport of amino acid and derivatives. So again, an interesting target to look at. Once again, we're establishing our initial conditions. This involves buffer compositions, any background to noise ratio, as well as testing your control sample, which is your untransfected parental cell line. In this case, you can see PAT1 and untransfected have a very decent window. And you can also look into differences between the members of a given family to compare transport kinetics, affinity, etc. So as I mentioned, PAT1 relies on the pH because it is proton-driven. So to test for the pH sweet spot, we divided our plate into 4 sections to test 4 different pH values. In each of those, we included our untransfected control every third column, just to make sure that the pH doesn't affect the membrane itself. So the untransfected parental cell line was added to the plate. And when we looked at the dose response of proline at different pH values, and we looked at the peak response, we didn't observe much of a difference. But when then we looked at the integral response, which is kind of like your area under the curve and correlates with the total charge translocation, we realized that pH 6.5 is producing the best looking curve quite consistently across different days tested. For that reason, pH 6.5 was found to be a sweet spot for the PAT1. We realized that the current observed at this pH is robust, and there was very little variability across the wells on different days tested, and we observed a good signal-to-noise ratio, as well as the untransfected was still not being affected at this pH. Having this validated and tested, we then rapidly were able to discriminate between endogenous substrates. In this case was L-proline and glycine on this graph. We observed outstanding reproducibility and correlation between tested days, as well as correlation between SURFE2R and SURFE2R96. And we managed to do fast compound characterization on the 96 well platform. Another interesting target that relies on pH is PEPT1, the human peptide transporter 1, which belongs to SLC15A family. It transports a wide range of things. It has a broad substrate specificity, and here are just a few examples of what this transporter translocates with the EC50s and kinetics determined on the SURFE2R platform. It translocates peptide-based drugs, ester products, artificial D and tripeptides as well. And for those reasons, it is being explored as a target for drug development and delivery, including in areas like anti-cancer and antiviral therapies. So definitely a very interesting target to look at, pharmacologically speaking. So of course we wanted to initially test how the pH affects PEPT1. And we noticed that pH influences the transport event. So here I'm showing you the overlaid traces from just one sensor, where the peak amplitude, which roughly corresponds to binding and translocation event, increases with the pH. So here in orange we have pH 5, and as you can see, as we increase the pH all the way to pH 7, the peak current increases. Another very interesting thing is that the decay, how fast the decay, which corresponds to the total charge translocated, decreases at each of the pH spectrum relative to pH 7. So let me break it down to you a little bit. What we did to visually aid this, we essentially moved the peak along its y-axis relative to pH 7. That's why this graph corresponds to the same peaks, but overlaid and stacked at pH 7. As you can see, at pH 5, the decay is pretty flat relative to pH 7. Similarly to alkaline pHs, when you have your pH 7 and then pH 9 being the decay slows down. Here I should also mention that the faster the decay, so the sharper the peak, the faster the transport, and vice versa. The flatter the peak kind of corresponds to the slowed down rate. With this, we can say that when pH is not optimal, less substrate is likely to be translocated. So we scale it up to 96 format to further investigate. And what we found out is that affinity of PEPT1 for gly-gly is determined by the pH. And the Km value for increases with the pH. And it goes from 1.2 millimolar at pH 6 to all the way up to 16.3 millimolar at pH 8. Because pH 6.7 was sitting kind of nicely in the middle, it was chosen for further studies and compound validation. So for our compound testing, we divided our plate into 3 sections to test 3 compounds in n equals 3, as well as including an untransfectant in every 4th column. And here I'm showing you a screenshot of the plate where we have highest concentration of the tested compound at the very top and reducing the concentration all the way down to your DMSO control. With 2 compounds, glibenclamide and zinc chloride, we observed a very consistent IC50 across 4 days tested. However, when we looked closely at the valacyclovir, we saw that at the highest concentration of the tested compound, we still observed about 30% of current. So here's just a screenshot of an exemplary well where we have a control current. This is just to remind you, the control current is only your DMSO, and it contains only your substrate that is translocated, in this case gly-gly. And here we have gly in presence of the full amount of the blocker. It was clear from this that we need to optimize the conditions for this particular blocker. So we looked into valacyclovir, and it acts as a competitive inhibitor. That means that it binds to the same side as the substrate gly. So we could first of all reduce the concentration of the gly from our regular EC80 to EC50 to improve the block. And also we know from previous slides that pH affects the apparent affinity for the substrate. So could it be relevant to inhibitors as well? To answer those questions, we tested valacyclovir in presence of gly at lower concentration, in this case 5 millimolar, at our standard pH 6.5, as well as increased pH 6.7. And what we noticed that by simply just decreasing the concentration of the our substrate gly, we are able to get full inhibition at the same concentration range. Interestingly, the same conditions but higher pH produced similar results to previous slide, meaning that we still had that 30% of current remained. So that just goes to show that both concentration of the substrate as well as the protonation state of the binding carrier have got an influence of the compound activity, in this particular case, valacyclovir. And this goes to show how important it is to test all the conditions and to determine the pH, especially for the targets that are heavily relying on it for its function. So just to recap, the SURFE2R and SLC research. Well, SURFE2R allows a huge assay flexibility. Assays parameters can be changed to find best working conditions without the need to use a new plate for each experiment. And buffer compositions can be easily changed to either test different substrates or pH values. However, SURFE2R is not just for membrane transporters. It presents itself as a unique opportunity to investigate lysosomal targets. And that is both for transporters as well as channels. If I've got people in the audience who have ever done lysosomal patch clamping, I'm sure you will know how challenging this technique is. Not only is very time consuming, it takes a lot of time to prepare your sample, and then once you finally have the sample, it's quite unstable, so your testing window is quite limited. To overcome that issue, oftentimes artificial system where you are overexpressing your target to localize into the plasma membrane are used. But this obviously, it's not the native environment of the given target that you're testing, so it might compromise the results to some extent. And with SURFE2R technology, we can use organelle fraction to be tested on SURFE2R to measure lysosomal targets. Here, again, it's just a schematic representation of the purification, where through the sucrose gradient, you're able to separate the sample for plasma membrane as well as organellar membrane and test your lysosomes using this. As I mentioned, and also Rocco mentioned, that you can store those samples indefinitely without a significant loss in signal, which yet again is another advantage of using SURFE2R for lysosomal targets. And now just very briefly, an example of a target that is expressed in lysosomes. In this case, I'm showing you TRPML1, which is a lysosomal channel. It is a non-selective cation channel, playing a crucial role in regulating lysosomal ion homeostasis. Defects in TRPML1 are, amongst others, lysosomal storage disorders. So first of all, let me convince you that the organellar fraction is indeed enriched in lysosomes and it contains them. And for that, we tested the crude sample, or your pre-sucrose purification sample, and organellar fraction with cathepsin B, which is a known lysosomal marker to confirm the enrichment of lysosomes in the organellar membrane fraction. Indeed, you can see that cathepsin activity increases and is distinctly higher in comparison to the crude sample, confirming that indeed sucrose purification successfully enriches the sample in lysosomes. So with that in mind, we went on and did a full assay validation on SURFE2R on trypamol-1. We assessed 2 compounds in 96SE format, and we saw excellent reproducibility. And what we also observed is a good platform-to-platform correlation, comparing results with fluorescence electrophysiology to our SURFE2R results. And now, lastly, I would just like to wrap it up with giving you an overview of our transporter toolkit. We can support your studies with our SURFE2R, which has very high sensitivity and low sample requirement for small studies or proof of principle studies. We can then easily transfer it to the 96SE format in the same way as I showed you today. It's a fully automated prep and run an analysis. We can also support your transport studies with flipper technology with high-speed kinetic readouts compatible with calcium and potassium and membrane potential assays, as well as synchropatch assays where we can fully control our environment too. Just a last note, I encourage you to visit our website and keep your eyes peeled. We have some exciting SLC related developments to share very soon. So yes, stay in touch. And with that, I would like to thank you all for tuning in and listening to my presentation. And I would like to thank Nanayan very much for inviting me to speak, to give a presentation about SURFE2R technology and how we utilize it at Sygnature Discovery. Thank you.
Thank you, Adriana, for your nice presentation. And I would say that we can just jump in with the questions. I see there are some. So I would start from the first two that I received from Carlos and YG. So I would read them together because they are quite related, I would say. So from Carlos is, what is the minimum number of vesicles that produce a signal? And from YG we have, during the spontaneous formation of liposomes, the number and volume of protein plus liposomes in each chip are different. Are capacitance and the amount of protein the main variables? If you want, I can answer this.
Yeah, go on. I guess you are the experts.
Okay, so there is not an easy answer to this, meaning it's not basically possible to say what's the minimum number of vesicles that produce a signal. That is because it depends on actually the speed of the transporter, or basically the turnover rate. So basically how much fast can the charge be accumulated inside the vesicle, and that depends on the protein itself. So there is not a general answer to this. Let's say as a rule of thumb, we have that for membrane vesicles, we tend to have concentrations around 0.1 milligram per milliliter total protein concentration. So usually we do a Bradford assay, we estimate the total protein concentration, and we see that this is kind of a sweet spot for having a signal. For proteoliposomes, we say usually to try lipid to protein ratios that are from, let's say, 5 to 20. But again, this is just a rule of thumb. It's not, it really depends on your target. Regarding the question from YG, related, of course, and sorry if I'm pronouncing your name badly. Yes, we do actually measure capacitance and resistance or conductance of the sensors. That is, of course, to ensure that we have a good sensor quality, hence that we have a good coverage of the SSM with our membrane. Because, of course, we don't want to have leak currents, or the other way around. We don't also want to have too much higher resistance, because it means that the sample goes too dense, and then, of course, the signal will be impaired. Okay, I go on chronologically. This, I think, Adriana, is more for you. A question from Manan. Hi, Manan. So in both cases presented here, EAA3 and PAT1, for the substrate affinity analysis, it could be seen that at low extracellular substrate concentrations, there was a small off-signal-like component at the beginning of the activation signal. Do you think that in such conditions the transporters are undergoing an efflux of ions?
Thank you for that question, Manan. So let me just read it again. For the off signal, so you mean after the on signal, the small. I think that really depends on the transporter. YET3 and PAT1 are, especially YET3, is relatively low turnover transporter. So the small off signal is not an uncommon thing to observe, especially when your on signal is quite small. I hope I'm answering your question. And yes, when your off signal happens, this is essentially what happens because the electrochemical gradient is being reestablished and those ions are being effluxed again because you're applying your non-activated solution again, and the conditions are being re-established, and you have the efflux of this ion out of the vesicle. So yes, this is essentially what happens. I'm not sure if you could add anything, Rocco. I guess that hopefully that answered that question.
Yes, I would say so. Okay, let's go on. A question from Francesco Tadini Bonisegni. Very nice talk. In the case of the proton coupled transporters, how did you generate and maintain the proton gradient? Did you also perform pH jumps on the SSM to investigate proton transport?
So in the case of the case studies I showed today were performed under symmetrical pH conditions. So for that, you essentially just need to rinse the plate or a single sensor with your new pH and re-establish that pH. So allow, let's say, 10-minute incubation, depending on the increments and delta between pHs, but up to 10 minutes is usually sufficient. In terms of pH jumps, yes, we have performed pH jumps, but not for those transporters. We have performed pH jumps where we try to establish the pH gradient for TMM175, as it was proved to be enhanced. The translocation of potassium was enhanced in the pH gradient, and we successfully managed to establish those conditions on SURFE2R, where fluidics are more separated between buffers. And for that, you simply, again, rinse your system with your resting solution and then apply your pH jump at a higher or lower pH, depending if you want to have inward directed pH gradient or outward directed pH gradient.
Okay, so let's go on. Next question is from Martin Glanthorpe. The success rate looks good. Do you generally find that all wells respond for optimized assays? What sort of throughput is achievable in terms of numbers of CRC per day for SLCs or TRPML1?
So that really depends on the transporter. For the ones that consistently work and there's a good signal to noise ratio and the assay window is relatively good, then success rate is between 95% to 100%. In terms of CRCs, with a well-optimized assay, we are able to drop it to n=2. And it depends if you'd like your untransfected present on the plate as well. That will change how many compounds you can fit on the plate. But usually an assay lasts an hour, depending on the incubation times as well. So within a day, we can run probably up to 6 plates, depending if the plate can be prepared the day before or the same day of testing, because there are some targets that we can do a day in advance. So yeah, depending on the numbers on the plate, anything between 5 to 8 plates a day we can easily run a day. In terms of SLCs and TRPML1, TRPML1 success rate is very good. It usually, again, is around 100%, 95%, and all the wells are pretty homogeneous. And again, similarly to how much a day we can run, similarly to what I said for SLCs.
Perfect. Thank you. So now we have a question from Wojtek Dvorak. Please bear with me if I'm pronouncing your name badly. Very nice data, congratulations. Have you ever tried also techniques such as Lysol-P for the organelle arrangement?
What, sorry?
Lysol-P.
Lysol-P. Thank you for that question and for kind comments. And not that I am aware of, not on SURFE2R anyway, from my perspective, but I know that we've had some internal development for the lysosomal batch clamping, and I'm not sure if they would have tried this.
Okay, then we have an anonymous question. How do you integrate SURFE2R results with other techniques you have in your transporter toolkit, like Flipper, TVC, or radio-labeled uptake assay?
So how we usually try integrate the SURFE2R technology. Well, for now you're just focusing on getting a good panel of transporters out there supported with SURFE2R technology. But what we would normally suggest is, if you are interested in a larger screen of compounds, we would go down the route of Flipper selectivity and then testing hit compounds on SURFE2R in a CRC format. But also we are able to do the hit screen on SURFE2R as well, single concentration n equals 2. And in terms of integrating SURFE2R results, we always compare our internal data with any targets that we've tested across the board. And some of them, I will admit, do not match. We had a case with TMM175 when we had compounds that did nothing. So it was a hit compound that we noticed was potentiating TMM175 in whole cells, but it wasn't doing anything in SURFE2R. And that was proved with a tool compound. I can't remember the name on top of my head, but it was proved that it's working through indirect pathways and it's activating TMM175 indirectly. And again, that could be another advantage of using SURFE2R. When you do your hit screens, you can select potentially the drugs that are working directly on your target versus the ones that might be working through indirect activation.
In fact, well, I'd say that this also answer actually another question, that is, can you share an example where SURFE2R allowed you to identify a hit or lead candidate that would have been missed with other platforms? We have another question. I guess this one, I can answer this. So are there any channel or transporters types that pose challenges for SSME on the SURFE2R? Well, yes, there are. So for channels, of course, the one that is not even a challenge, is that you cannot measure them, are voltage gated channels, because of course it means you need voltage just to open them. And SURFE2R starts its measurement with a voltage of 0. So if you need that to open the channel, then the channel will not open. You will not be able to measure anything. Regarding transporters, yes, they can be too slow. So as of now, we think that, actually we empirically tested that a turnover rate down to 0.5 seconds to the -1 is the minimum possible to achieve some kind of signal. Below that, it's very hard. Of course, you can help by basically having more protein. So you enrich of sample, and that should help by incrementing the signal. But of course, it's all empirical. You just need to test these things.
Also, so if I may add,
sure, of course.
Additional thing to answer that question is that you cannot measure transporters that do not produce a net charge translocation. So if you have exchangers that produce a net charge of 0 because you have one sodium going in and one potassium going out, then essentially you will not be able to measure any meaningful charge translocation on SURFE2R platform. But for that's why we try and integrate other platforms to complement the SLC research.
I would integrate your question by saying that in the case of exchanger, it's theoretically possible to see half reactions. So in that case, basically, you could see, let's say, a potassium proton exchanger. You could do pH jumps in absence of potassium, and you could be able to see a net proton flux, but that would be only alpha reaction. If you, of course, if you are interested in the whole transport, as you correctly say, an electroneutral exchanger is not a good target.
What if the affinity for one of the ions is higher? Would that dominate, or again, would be potentially?
It's more about the speed in the end. So basically, let's say that hypothetically we are in the condition of having a overall electroneutral exchanger, but the semi reaction, they are very separate in time. Then hypothetically you would see something like this, basically, like on
Yeah.
like a peak
A
of
negative and
something.
then positive followed by a positive, yeah.
Exactly. But to date, I never see something like that. But theoretically, yeah, it's a possibility. Okay, so I see no more questions. Actually, I ask you one, a bit on a general, this very general thing. So how so working for a CRO, of course you have I am guessing quite high standards that you want to keep. So for you, how do you decide when an assay is robust, basically?
Well, multiple things come to play.
Mm.
So for a robust assay, you obviously want your services to sell, so you don't want it to be overpriced. So again, that's why we looked into different expression methods to see if we can enrich the sample, as you mentioned, answering a previous question, to stretch the sample more. That's one thing. Obviously, reproducibility, assay to assay and day to day. Then another thing is how homogeneous the signal is across the wells. If there's any edge effect, how it responds to the compounds, again, reproducibility. If it's, you can relate the data to literature, or if there's no available, obviously, you do everything that you can with what you have. So I guess those are probably the main points to consider the assay robust. Definitely reproducibility across days and variability in terms of response. Those are the main things.
Okay, thanks. So anyway, it's quite challenging overall. Okay, yeah, I see no other question in the meantime, so I would give back the word to Jaimar.
Yes, so just very shortly, I wanted to thank Adriana and also Rocco. It was a very nice discussion. Also, thanks to all of you who put the questions and who write also the comments. Yeah, it's always very nice to have a lively discussion and yeah, to have your participation. Before we go, just very quickly, because yeah, since you are here in this webinar, regarding SURFE2R, the SURFE2R platform. So you might be also interested in attending another webinar we will have in September, 24th of September, where we will be talking about unlocking the full potential of the SURFE2R data. So this would be more on advanced analysis techniques. So you are all invited to join us there as well. So you can yeah, go to our website under events and then register there. Yes, and if you are around Europe or in Germany in October, you are also very welcome to join us on October 28th and 29th for the Nanion user meeting. So it would be very interesting scientific talks, poster sessions, and of course some networking sessions. So yeah, if you're around, we would be happy to see you here. But if you're also not around, you can join us also online. So yeah, there are these two modalities. So you will not miss anything from the very interesting talks that we will have there as well. So that's it for today. Thank you very much for participating. And yeah, we will see you there in the next webinar, in the next event. So take care and have a nice day.